Welcome everybody. Välkommen allesammans. Jag snackar lite norsk, but I will continue in English. So, welcome everybody and good afternoon, but also good morning for our Canadian speakers. First of all, thank you for attending to our webinar about the effect of seafood consumption on the brain. My name is Rachel Durant, and I am the project manager of Marine for Health for NCU Blue Legacy. Blue Legacy is a cluster for innovation and value creation based on marine resources. And the aim of the cluster are to promote sustainable innovation and profitable production of marine ingredients from various marine raw materials. The Marine for Health project purpose is to establish competencies and scientific expertise in health effects from marine resources. Before to start, just a reminder, this webinar will be recorded, so please mute your microphone and turn off your camera. You can also ask your question directly in the chat, and we will answer it during the question session at the end of the webinar. So today, three different speakers are going to present their work about seafood and brain health. So we will start the webinar with Lisa Colden-Midtbø. She has a master's degree in human nutrition and a PhD in seafood and health effects. She currently works as a researcher at Møller-Forskning, and she will present results from studies performed at Institute of Marine Research in Bergen. Next, we will have a presentation from Frédéric Callon from the Laval University. Is a professor at the Faculty of Pharmacy in neuropharmacology, neurodegenerative diseases, and the blood-brain barrier. His team mainly performs preclinical assays testing of nutraceutical and pharmaceutical in animal models, as well as neuropathological studies on human brain samples. And finally, we will have a presentation from Lucie Beaulieu. She's a professor at Laval University at the Food Science Department, and she initiated the research program on the structural and functional characterization of new sources of marine ingredients. She is also the coordinator of the interest group on biomarine products at the Institute of Nutrition and Functional Food, INAF, in Quebec. So now I will give the word to you, Lisa, and please you can share your screen with us.
Thank you for the nice introduction, Rachel. Let's see here. I will now try to share my presentation. Let's see if...
Yeah, you can put it in full screen.
Yeah, I am. It's slow. Can you see the presentation now?
Yeah, we have the notes. But,
oh, notes again. 1 second.
Yeah.
Okay.
Yeah.
Perfect. So I'm Lisa. I'm a researcher at Møller-Forskning. Møller-Forskning is a research institute located on the west coast of Norway, in Ålesund, Molde, and Kristiansund. And we have a close collaboration with the marine industry and work with the whole value chain from raw material to end products. And my research area is mostly seafood and health effects. However, today I will present a study performed at the Institute of Marine Research in Bergen, as Rachel said, where I used to work before I started in Møller-Forskning. And this is a randomized intervention trial on preschool children in Bergen, performed in 2015. And Janneke Øyen was the project leader. However, I was involved in all the steps in this study. And we have published the results from the study in BMC Medicine in 2018. So a background for the study. It is well known that fatty fish is rich in omega-3, vitamin D, and protein. And meat is a good source for protein and iron, among many things. So both fish and meat has positive components. And we have omega-3 fatty acids in the brain. DHA is dominant omega-3 fatty acid here. So between the brain cells, there are millions of messages sent every second. And the theory is that this job is performed easier if it's enough omega-3 fatty acids in the brain. And also vitamin D and iodine might be positive for brain development, especially in children. In observational studies, there has been a positive correlation between seafood intake and cognitive function, but results from randomized intervention trials of individual components like omega-3 capsules show inconclusive results. So when this study was conducted, it has not been performed intervention studies with seafood on children. So the main aim for the study was to investigate if intake of herring and mackerel improved cognitive performance in 4 to 6-year-old preschool children compared to intake of meat. And we did this by using an IQ test called the Wechsler Preschool Primary Intelligence Scale. I would just call it WIPSI, and motor skills by 9-hole peg test. Yeah. And this is the experimental setup. The recruitment of kindergartens in Bergen was performed in November 2014. And the data collection started in January 2015. And it was over a period of 6 weeks because of the workload. We wouldn't be able to do all the data collection at the same time. And the intervention lasted for 16 weeks. And also the data collection after the intervention were spread over 6 weeks period. And the 2 interventions group were fish with herring and mackerel, and meat with chicken, lamb, and cattle. And they had this for lunch 3 times per week for 16 weeks. We had to increase the amount of fish and meat because the children wanted more than we anticipated. So it was popular for lunch. This looks quite complex, but it's just a typical overview of the recruitment of children and how many that participated. So 17 kindergartens were invited and 13 kindergartens agreed to participate. And in these 13 kindergartens, there were 314 children between the age of 4 to 6-year-old. And only 232 agreed to participate. And we lost some to follow up and some decline from the study. So at the end, it was 118 divided into 2 groups of children that did the pre and post cognitive tests. And the composition of the meals, here we have meat with a yellow line and then herring and mackerel. And of course, the herring and mackerel is higher in EPA and DHA. However, the catering company used fried the food, some of it, to make it more interesting for the kids. So we were interested to see the omega-6 and omega-3, but still after frying the herring, the fish balls and fish sticks, it was still much higher EPA and DHA. And iodine was also higher in the fish meals. Vitamin D were not that high, as we expected higher levels. For tests, we did, like I said earlier, VIPSI test for concentration, memory, and language, and motor skills with 9-hole peg test, the blue one on the picture. And we had questionnaires for food, mental health, and socioeconomic status. We took some biological samples from the brave kids before and after the intervention, both blood samples for vitamin D, fatty acids, and iron status. I used ferritin in the later slides. We did hair samples for mercury level, just for safety, and urine samples for iodine status. And this is a big table, but it's just the baseline characteristics, just to see if there's a difference before we start the intervention. And in Norway, it's recommended to eat fish 2 to 3 times per week, and 47% of the parents reported that they ate the recommended level. So the kids had a high level already of fish. We tried to recruit from different social groups in society, but as you can see, we didn't succeed that much. But this doesn't show the amount; it's just the frequency. So we don't know how much they ate for each meal. And here, the p-value has to be below 0.05 to be significant. So nothing was significantly different between the groups for age, weight, height, education. Yeah, they were quite equal before we started. So the biological parameters, what we analyzed. This is also a big table, but I will guide you through it. So AA, LA, it's omega-6, and we saw a small increase in the meat group and a small decrease in the fish group, but it's not a big change. For EPA and DHA, which is omega-3, we saw no change in the meat group and a change to the higher level in the fish group after the intervention. And ferritin, which is iron, we saw an increase, but not significant in the meat group, and a decrease, significant decrease in the fish group. So this, you have both beneficial results and negative results from giving them fish for lunch 3 times a week. Both groups were reduced in iodine, which was interesting, but lean fish is a good source for iodine, and this was fatty fish. So maybe we replaced something they ate for lunch so that the iodine were lower after the intervention. But the decrease were lower in the fish group compared to meat group. For the vitamin D, we see that they had a good status before the intervention. You see on the left side, it's the meat group before and after. And 50 is a sufficient level for vitamin D. And they had a mean level of 60. So they had a good level before, and they both increased in vitamin D. And this was mostly because of sun exposure on holidays to southern countries, or as we call it in Norway, they went to Syden. So that's the main reason for the increase in vitamin D and not the fish. However, the omega-6 index, it's omega-3 index, sorry. It's the EPA and DHA of all the fatty acids in the blood. And they say that for adults below 4%, it's increased risk of cardiovascular disease, and if it's higher than 8, it's a protection against cardiovascular disease. So they increased, the fish group increased more after the intervention with the fatty fish. So this is nice results from the study. For the results on the motor skills, we saw that the fish group had a slightly better improvement for the non-dominant hand than the meat group, and no effect for the dominant hand. So yeah, it's not the most convincing results. And the VIPSI test, you see the green and the blue here, they had no improvement in the IQ test. However, an interesting point is that the fish group ate less. They ate 47 grams per meal compared to meat, they ate 61 grams per meal. So if you adjust for the compliance, the eaten fish, it is significantly better results on the IQ test for the fish group compared to the meat. And as we see here, this is a graph showing the fish in blue and meat in red. And on the bottom line here, it's food consumed. And on the vertical line, it's change in the total IQ of the test. And we see that the more they eat of the fish, the better they result on the IQ test. And we don't see the same for the meat group. So the conclusion is that there was no significant difference between the intervention group on cognitive performance from pre to post intervention in crude analysis. The EPA and DHA status increased in the fish group, and the fish group had significant larger improvement on the IQ test compared to meat group after adjustments. And this is acknowledgements to the participating children and their parents. And we collaborated with psychologists at RKBU. And thank you for your attention.
Thank you, Lisa, for your presentation. It was really interesting. So now we move on to the Frederick presentation. Please.
You might need to help me to stop the sharing.
Yeah, it's okay.
Thank you.
And please reminder to use the chat if you have some question for Lisa. Please, Frederick.
Yes. Okay, I was looking for the button for the sound, so it's fine now. So you see my slides? Okay.
Yeah, we see it.
Good. So thank you, Rachel, for the nice invitation. So I'm gonna talk to you a little bit today about some data out there from our lab, but also mostly from different groups in the world that show some evidence of health benefit of omega-3 fatty acid, particularly for the brain. So I'm located in Quebec City, Canada, and I'm a co-director of the UPCHI NutriBrain Lab, which is an international lab with Sophie Laillé in Bordeaux in France, in which we study links between diet and the brain. I'm also, as Rachel said, a professor at the Faculty of Pharmacy at Laval University. Okay. So today I'm going to talk to you about some take-home messages about omega-3 fatty acids, how they are present in the brain, how they reach the brain, and how the diet can influence the brain content. Then after that, I will show you some evidence of association between omega-3 fatty acid and Alzheimer or Parkinson diseases. Then a few words on mechanism, and a conclusion at the end to show you that now omega-3 fatty acids are considered as a key part in most ongoing intervention studies in Alzheimer's disease mostly. So I won't go into all the details, but the omega-3 fatty acid we are mostly interested in are EPA and DHA, which come from marine sources. And I won't talk so much about omega-3 fatty acid found in vegetable oil like alpha-linolenic acid. So it's mostly about DHA and EPA. And one of the reasons is because you find a lot of DHA in the brain. So the first thing I think important to know is that the brain is full of fat, okay? The human brain, there's a lot of fat in the brain, so it's 50% of dry weight. So that's a lot. And within the fat in the brain, omega-3 fatty acid stands as one of the most numerous fatty acid you can find in the brain. So among all fatty acid, it's about 15 to 20%, which is DHA actually, and phospholipids in the cellular membrane. So that's a lot. It has to be important, right? And what's fascinating and has been shown in the last, I don't know, maybe 30 years, is that brain lipids will be influenced by the diet. So when you eat carbohydrates, most of it will be burned. When you eat proteins, some of it will go to build proteins in the body, but most will be changed in some way. But among the lipids, of course, a big part will be burned, but there's a fraction of the lipids that will end up in the body, in cellular membrane, pretty much as it was ingested. And in the brain, I will show you a few graphs of the measurement we've made in animal models. So what I show here, these are non-transgenic mice compared to transgenic mice. These transgenics are actually animal models of Alzheimer's disease neuropathology. But this is really not the point. The point is really about what we give them to eat. And mice, they are not free, so they basically eat what we give them. They have no choice. So when you give to a mouse a lot of DHA, we see a very nice increase in the brain. This is true for all these two animal models. So if you look here, I don't know if you see my pointer, but there's almost no overlap between the animals fed with a high DHA diet and those fed with no DHA. And what's also interesting is that we do see an increase of EPA as well, because there's some retroconversion from DHA to EPA in the brains of these animals. So you can get an increase of about 2 to 3% in the brain. And it's even more obvious when you feed the animals with EPA. So, well, I mean, yeah, it's even more obvious when you measure EPA in the brain of the animals. So when you give them a high DHA diet, you see the increase of EPA in both models. But if you feed the animals with a high EPA diet, then you see an even clearer increase of EPA. But what's important is that the basic level of EPA is really low in most, I would say, in a mouse brain when it's fed a low omega-3 fatty acid diet. So it's pretty clear that you can change the levels of DHA and EPA in the brain with the diet. And this is not only for omega-3 fatty acids. When we feed the animals with a canola oil diet, a lot enriched in MUFA, monounsaturated fatty acid like oleic acid, you can also see an increase in the brain. So it's really a general thing that we can say is that the lipids in your diet, we have an influence on the concentration in the brain, and it's even more clear for DHA. So how do omega-3 fatty acids enter the brain? Most evidence suggests that they can freely cross the BBB by passive diffusion. If you look at the structure, I don't know if there are chemists out there, but it's really a carboxy group with a lot of carbon. So this is highly lipophilic, so it should not come as a surprise that it can cross barriers, and including the blood-brain barrier, and then reach the brain. And there are many evidence beside the chemical structure. There has been some studies that indicate that DHA, free DHA in the blood can supply the brain and provide a fast supply of DHA for the brain. And this uptake is not saturable. I will show you some data about that. So in the lab, we do a technique called in situ cerebral perfusion, in which we inject a compound directly into the carotid. Then we take out the brain, and we can do relatively simple calculations with that. And what we show is that when you give, by this method, radiolabeled DHA or radiolabeled EPA, we can see an increase over time in the brain. And we get these kind of numbers around 50 microliter per grams and per second. These kind of numbers are actually pretty much the same as you can get with highly lipophilic drug like Valium, for example. So it shows that DHA and EPA can readily cross the blood-brain barrier. And this is what we call a competition experiment. So when we increase the amount of DHA, if there would be a receptor that could be saturated, we would see normally a decrease if we increase the concentration. But in that case, it stays the same. So what it shows is that there's no saturation. So even if you have more DHA, it will cross more the blood-brain barrier because it's mostly by simple diffusion. But just to say that this is not to say that there's no mechanism of regulation. There are. I won't have time to go into that, but there are some mechanism to control the transport of omega-3 fatty acid into the brain. And I think a point that I think is pretty well accepted now is that the consumption of omega-3 fatty acid is now at its lowest point, I mean, in the history of humanity. We were probably more exposed to a higher ratio omega-3 to omega-6 fatty acid when we were hunter-gatherer. With the advent of agriculture, we started to select crop with a low amount of omega-3 fatty acids, and with the processed food during the industrialization, this amount went lower. So we can think that there are many people out there, maybe less in Norway, I don't know, but let's say in Texas, more people with a lack of omega-3 fatty acids, and this maybe contribute to our evolution. The thing is that we were selected back then, 100,000 years ago. So our genes are adapted to a relatively high level of omega-3 fatty acid consumption. So a few words about Alzheimer's disease. I won't talk to you much about the disease. I can answer questions about that. I will just focus on some of the data with omega-3 fatty acids and cognition. So there are preclinical studies in animal models. The vast majority of them show cognitive benefits of DHA in animal models of Alzheimer's disease. There is also stronger and stronger epidemiology evidence showing that higher consumption of omega-3 is associated with a lower risk of dementia or Alzheimer's disease. And there are also some very large studies indicated that blood DHA concentration is in generally a lower, a lower blood DHA concentration is associated with a higher risk of Alzheimer's disease. And this results from a meta-analysis. So the levels of DHA in the brain is one of the strongest indicator of a higher risk of developing dementia or Alzheimer's disease. However, intervention studies in general have been less positive, especially in Alzheimer's disease, probably because it's too late. But for the intervention that were at earlier phases, before the diagnosis, some of them were actually positive. But I won't go into all the details. I can answer more later on. It's not clear if omega-3 fatty acid has an effect on the neuropathology, on the progression of the disease, the actual disease in the brain. There are very few evidence in humans because, of course, you cannot really look into the brain except by imaging. So some MRI data suggest that you can prevent a little bit of volume loss with EHA and DHA, but it's not necessarily very strong. Even in animal models, there's some evidence, but there's some conflicting data as well. But still, we can hope that maybe there's some modification of the disease occurring there. So in summary, we have strong animal data, epidemiology data that suggests cognitive benefits. For the neuropathology is not so clear. The clinical trials have been negative in Alzheimer's disease, but probably because it's too late. So it's really about, the idea is really more an objective of prevention, I would say. This can be modulated by ApoE, genetic status and dietary status. So this is a big factor in all these studies that it's hard to get rid of. And we have also evidence that saturated fat can be actually bad. So it's not all fats that are good. It seems to be more omega-3 fatty acids and other types of fat. Saturated fat seems to have more negative effect. There are also some data in Parkinson's disease. I won't go into all the details, but we've done some preclinical work showing that you can have neuroprotective effect before the diagnosis and some neuro recovery after the diagnosis. There's a big lack, I would say, of epidemiology in Parkinson. There's some studies, but not so much. And there's no really good clinical trial that has been done in Parkinson's disease to investigate that hypothesis. But I think there's some hope in Parkinson's disease as well for a role of omega-3 fatty acids. For the mechanism, I will be happy to answer questions about that. The only thing I can say is that if you can increase the amount of DHA in the brain, it means that the DHA will get into the membrane of the cells, and from there can change the function of the cells and can change also the different pools of omega-3 fatty acid you will find in the brain, can change the level of different active metabolites, and all that can have an effect in the brain. But like I say here too, the effect can be also through the periphery. So omega-3 fatty acid can have an effect on cardiovascular system, metabolism, maybe the microbiota, and from there have an effect on the brain as well. So on this slide, what I want to show is that things that, I mean, the general mentality has changed in the field of Alzheimer's disease. I think people agree for a long time that environmental factors are important, but it's relatively new that people are accepting that nutrition can be one of these modifiable risk factor of Alzheimer's disease and Parkinson's disease in a lower scale. So this is now widely accepted. So I will just finish by looking at what are these modifiable risk factor of Alzheimer's disease. So of course, education and age, well, I mean, age, no. Education, perhaps a little, genes, not so much, ApoE, not so much. Cardiovascular risk and diabetes, probably in part by a control of the disease. Sex, no, but nutrition and exercise obviously can be places when we can intervene. And what's interesting is that diabetes, hypertension, and obesity in midlife are now well accepted as risk factor of Alzheimer's disease. But as you know, the diet can affect all these risk factors. So even if it's difficult to isolate the diet as a variable, we know that diet will have an effect on very important variables and risk factors for Alzheimer's disease. So, and the good news based on the population-based study is that it has been estimated that about 35% of AD cases could be prevented by controlling these different modifiable risk factors. I won't go necessarily all through them, but as I just said, diet can have an effect on many of these risk factors of Alzheimer's disease. However, when we perform intervention, the picture is less clear. So there has been 3 very large multi-domain lifestyle intervention studies, like one in Finland, one in France, and one in Netherlands. The one in Finland, the finger was considered relatively positive, but the two others, not so much. But I won't go into all the specifics of all these studies, but of course they had the different primary versus secondary endpoints. So it's promising, but it's still a bit conflicting. But still, in the field of Alzheimer's disease, a big chunk of investment is going into what they call the worldwide fingers. So these are these kind of large studies in which the multi-domain intervention studies in which the lifestyle is controlled for a certain group of patients. So, but this includes the diet, but also exercise, control of cardiovascular factors, risk factors, and other interventions. So in many places in the world, there are all these kind of studies now going on. So the point I want to make is that it's getting really accepted in the field of Alzheimer's disease, and there are really big, large studies going on as we speak, and maybe in a few years we will get more information about that, and diet is part of it. So just to summarize what I've just said, so the brain is made with a lot of fat, a lot of omega-3 fatty acids. Omega-3 fatty acids can cross the blood-brain barrier and supply the brain, because sometimes there are some, I would say, people that question this, but this is pretty clear in our hands, at least. And so all this indicates that omega-3 fatty acids are essential for the function of the brain and can be modified by the diet. And they have some evidence of possible benefits against neurodegenerative diseases. For Alzheimer's disease, strong epidemiology, some studies in animal model. Randomized clinical trials have been less clear. For Parkinson's disease, we have a little epidemiology, strong preclinical studies, but we're still lacking good clinical trials. And the last point is really that they are widely accepted as a key part of ongoing multi-domain intervention studies in the field of Alzheimer's disease. And finally, it's more about prevention versus treatment. So that's a little bit the bad news. If you have somebody or a family member that come and talk to you and ask about taking more omega-3 fatty acids, unfortunately, if the diagnosis has been made, it's probably relatively too late. There might be more hope in Parkinson's disease when the diagnosis is made, but in general, I would say it's more about prevention than treatment. So I've been working with many different support agencies in Canada, and thank you for your attention.
Thank you very much, Frederick. So many information. Now, Lucy, if you can share your screen.
Yes.
Yeah.
Okay, it's okay? Because I have two screens too, so it's okay?
Yeah, it's perfect. Thank you.
Okay, good. So first, I would like to thank the NCE Blue Legacy for giving me the opportunity to present this new project entitled Neuronalg, the beneficial effects of St. Lawrence seaweeds on cognitive health. This multidisciplinary project include the researchers from the Institute of Nutrition and Functional Foods, so Dr. Calon, Marette, Tamino, and myself. And also industrial partners are involved, such as Salaweg, a seaweed farming producer, and Innovactive, a health ingredient supplier, and Marinov, a college from the Gaspé Coast in Quebec. So through this project, we also have a collaboration with the International Associated Laboratory OptiNutriBrain. And the project is supported by the Quebec Maritime Network and the Quebec Research Funds. Okay, so change, okay. So first of all, I just want to give you a few words about the Institute of Nutrition and Functional Foods. This is a strategic group supported by the Quebec Research Funds, And it represents the largest group of researchers in Canada dedicated completely to foods and the interactions between food, component, nutrition, and health. So INAF encompasses 90 researchers, 14 academic organizations, including industries, not industries, universities and colleges. And it counts more than 400 graduate students and postdoctoral fellows. And also it counts more than 200 partners. Okay. And there's an interest group on marine products and byproducts, and I'm the coordinator of this interest group for 12 years now. And very recently, now I'm director of the team Resources, Marine Energies, and Health of the Maritime Economic Sector of the Quebec Maritime Network. And several topics are studied through the interest group on marine products. So research interests include valorization of biomass by the production, purification, and characterization of target molecules. We also have screening for antimicrobials or demonstrating the health benefits of marine ingredients. Oops. Okay. And among the marine products, marine algae are an attractive source of nutrients and active compounds. And the chemical composition of algae is variable according to many factors, such as the species, season, geographic distribution, cultivation parameters, and processing. And since 10 years, there has been a production increase of 8% per year of marine algae globally, with a global market of $6.4 billion US. So marine algae compounds are used mainly for the food ingredient industry as sources of polysaccharides having texturing properties such as carrageenan. And also algae are traditionally consumed in Asia, as you know, as sea vegetables. We just have to think about sushi. And they have been recently promoted in the cuisine of several European and American countries. Oops, sorry for my animation is late. And Canada is the longest coastline in the world and is an important source of biodiversity. In this project, we study marine algae located in the north of the St. Lawrence River and in the Gulf of St. Lawrence. So I don't know if you see here, and Norway is here. And marine algae are grouped under 3 phyla: brown, red, green, based on their pigmentation. And here are some examples of edible seaweeds, such as Sicurina, it is called kombu, or Palmyra, the dulse, and sea lettuce, the ulva. In Quebec, the seaweed supplies are mainly based on the harvesting from wild and the cultivation with long line method on marine farms. There's also a cultivation in water tanks, but it is still in the experimental stage. Okay, it does not currently allow the production of marketed seaweeds, but it is interesting to study the effect of cultivation conditions on the biological activity. For instance, our previous results suggest that tank cultivation of seaweeds is a promising tool to control algal physiology and then enhance the activity of targeted bioactive compounds. And marine algae harvested or cultivated are under investigation for functional food applications in Quebec. However, no data are available on the beneficial effects of Quebec marine algae on cognitive health. So that's the reason of our project. So we would like to have interesting seaweeds for the brain health. And marine algae represent a very diverse pool of neuroactive compounds, such as polyphenols, such as fluorotannins, sulfated polysaccharides, sodium oligomanate, proteins and amino acids, and carotenoids. For examples, polyphenols are studied in Alzheimer's disease for their abilities to scavenge free radicals to reduce oxidative stress, which may in turn lower subsequent inflammation and reduce risk of cognition decline. And recently, GV971, a pharmaceutical product derived from brown seaweeds and composed of sodium oligomanate, was approved by Chinese agencies for the treatment of Alzheimer's disease. And GV971 is thought to attenuate neuroinflammation by modulating the intestinal flora. And also another example, it's microalgae. It contains a synergistic complex of molecules, including some typical amino acids. And these amino acids have the potential to cross the blood-brain barrier via amino acid transporters. And according to in vitro studies, it inhibits monoamine oxidase B, an enzyme involved in the metabolisms of monoaminergic neurotransmitters. And these molecules have different mechanisms of action and different impact on brain health. It is better known by my colleague, Frédéric. And the general objective of the neuronal project, and it's a really new project, sorry, because I cannot see, okay. It is to produce scientific knowledge necessary for the development of extracts enriched with neuroactive compounds for the production of bioactive ingredients from marine algae from St. Lawrence in Quebec. And there are specific objectives for this neuronal project. The first one is to establish optimal cultivation conditions to stimulate the synthesis of neuroprotective compounds in red algae. It will be performed by our colleague at Marineuve in the Gaspé Coast. The second objective is to develop methods for extracting and characterizing various extracts of algae enriched with potential neuroprotective compounds. So we will work with Innovactive too for this objective. And also for the third objective, we want to assess the bioavailability of these biomolecules and their potential effects on brain health in animal models, but focusing on the microbiota gut-brain axis. The first objective aims to support the stabilization of supply chains in the context of industrial exploitation of cultivated red algae, since these have never been the subject of sufficient effort to allow commercial exploitation. This project plans to work on the cultivation of red algae in marine farms with the fluctuating cultivation conditions, and it's with a lower cost. And also we want to work with tanks, so water tanks, with controlled cultivation condition, but it's a higher cost for this experiment. And according to the results, so we would make the best choice for further seaweed exploitation. In the second objective, the production of both brown and red algae extract by Innovactive, our partner, will aim to enrich water extracts with these molecules of interest, which is more in line with commercial expectations. We don't want to use a lot of solvents, so we will use water extraction. And the analysis of the compounds will be carried out by HPLC, and the identification of active molecules will be performed by LCMS, so mass spectrometry. And the methods for the identification of algal compounds will be used for the analysis of biological tissues collected from animal models in Objective 3. And in the third objective, we will analyze the systemic and cerebral bioavailability of extracts in mice. Bioavailability will be measured in plasma, brain, feces, urine, liver, fat, and lung. Basic toxicity analysis will be performed in these animals, and feces collected at the beginning and at the end of this protocol will allow us to study the relative abundance of bacterial population in the gut microbiota. Metabolomic analysis will be also performed to follow the impact of the extract on the modulation of microbiota. And what we expected from this project is first to scale up the production of extracts enriched with molecules having neuroprotective effects. And also we want to generate knowledge to better understand how the modulation of the microbiota or its neurometabolites can contribute to the prevention of diseases affecting brain function. And we also expect to improve interactions between the fields of aquaculture, marine biotechnology, and the nutrition health sector. So it's a very stimulating project and challenging project too. So I would like to thank you for your attention. Tax Kaldua.
Tusen tack, Lucy. Thank very much for this, for all your presentation. So now we are going to, now we are going to respond some question. I have one question for you, Lisa. About fatty fish are mainly associated to different toxins and also to heavy metal concentration. And in your presentation, you present that you analyze the content of heavy metal. Do you have some comments about that, how it can impact the children's health?
Well, they have seen that in animal studies that different heavy metals and PCBs and other undesirables might have a negative effect. However, in this study, I didn't show that, but the levels were way below the set standard levels. So, and also the mercury, it's the fatty fish isn't, it's not that high mercury levels in fatty fish as in lean fish. So they were low compared to what the standards are.
Okay, and so do you think, because do you think that the impact will be the same for children, for the brain, if they will eat white fish than fatty fish?
Well, it's a very different components. So it's higher in iodine with lean fish, but lower in omega-3. However, the omega-3 is often in phospholipids, and we have seen in animal studies that the uptake of omega-3 is better from phospholipids. So in the tissue, we see a higher level than expected. So it's very beneficial with lean fish also. That's why we the government recommend both lean and fatty fish. But for omega-3, it's a bit higher levels in fatty fish. So they say, I think it's 2 out of the 3 meals should be fatty fish.
Okay.
At least in Norway.
Okay, so especially for the concentration of omega-3 and iodine.
Yes, and vitamin D, and it's a lot of beneficial components in fish for not only the brain but heart and everything.
Frederick, I think we have a question for you from the attendees. So if a person have had a bad diet for many years and then start with a good diet with a lot of food and exercise, how long will it take to reach a perfect and healthy level of EPA and DHA in the brain to prevent diseases?
Well, I don't think we have an exact answer for that.
Yeah.
Because obviously we cannot perform that kind of study with the human brain. From what we see in animal models, it's not clear exactly the timeline necessary. But what I would say is that I think there's always hope. I mean, it's never too late, basically. I think you can still start at any age. And I would expect within a few months, you can get some of the impact on the brain. So some of the effects can be probably relatively fast when you talk about incorporation into cell membrane. Probably occurs relatively fast. So I would say that yeah, it's never too late.
So that means that it's better if you start to eat fish or to take omega-3 when you're young.
Well, it's always better to start early. Yeah, obviously.
We have a question for you, Lucy. So it's about algae, of course. What is the current price per kilo for the different marine algae for the market in Asia? And is the market there is big enough to handle big volume of farm algae from Europe and from America?
Oh, it's a big question. The price for a kilo of seaweed from Asia, or I know for in Quebec, because it's a very new market, when we have dry seaweeds, 1 kilo, it costs maybe $20.
Mm.
And the other part of the question was?
The market there is big enough to handle big volumes of farm algae from Europe and from America. Can we compete the market algae?
Here in Canada, in America, we start to cultivated seaweeds. Of course, it's a big market in Asia, but they are facing some problems with pollution too, and also space for the marine farms in the ocean. So I think we have a place to take here. Okay. But I don't know if I answer well to the question.
No, it's okay. What? No, it's okay. It's perfect. Sorry. I have a question also, Frederick, for you. I don't know if you read the last GOED meta-analysis about omega-3 and DHA, and they talk about the importance of the dosage. Sorry. What do you think about there is a dosage for to have an impact in the brain health? At least.
Well, it's a bit. Yeah, the dose is a complicated question because you can have various range of doses that you can use. Mm-hmm. There's probably a dose to prevent, well, I would say to avoid depletion. So that dose is probably low. So probably 2 servings of fish per week is probably enough to make sure you're not in a state of deficiency. But you can also go up to more pharmacological doses like 1 or 2 grams per day. The thing is that there's not so much adverse effect with omega-3 fatty acids if you increase the dose. So for some studies, they have used those that are relatively high. So for example, for some studies in the cardiovascular field, they will be at 1 gram per day or more. But in practice, this becomes a bit difficult if you want to get it from fish, right?
Yeah.
So that's also the other question. Do you want to go with fish or do you want to go with supplement? I think in general, fish is probably better because there's also other nutrients and it's part, of a way of living, more a lifestyle thing. If you go only with supplement, maybe you can miss some other things, but at the same time, if you want to really increase the dose, you have to go with supplement. There was some patient that were contacted in me too, because they wanted to apply this patient with Parkinson's disease, and there was a person, he wanted, I think, to eat one of these big cans every day. I don't remember if it was mackerel or tuna, but tuna is maybe easier, but like salmon, you can get bored a little bit every day. So it might be difficult with food to reach these higher doses.
Actually, it was one of my other questions for you and Lisa also. You can also tweet. What is the difference between between to take food supplements or to eat directly fish? There is some difference. So it was, because as you say, Frederick, it's pretty difficult to reach high omega-3 dose if you eat only fish. So there is a different impact.
Well, I have a comment there, because when you eat fish, you replace maybe a burger with the fish, so you have better balance with omega-3 and omega-6. But if you just take 2 capsules and go to McDonald's, you don't have the same effect. So if you look at it like that, it's better with a fish meal.
Okay.
Yes, I agree.
Yeah.
It's a problem here in North America.
Yeah.
Some people
I guess in Norway also.
They will not change the diet, but they feel better to take a supplement and they say, oh, I'm okay, but maybe not.
I'm healthy.
Rachel, maybe I have a question too. It's just because for Lisa, in your study, we focus on omega-3, but we know that proteins are known to have a good impact on the cardiovascular disease. So can we think we have a synergy between fish proteins and omega-3? Or?
It's a good point. We tested the different components, and DHA had the same effect as on the, I think it was the VIPSI. So we think it's mainly DHA, but it's complex, a meal. It can be components working together. So we need to study it more to be sure that it is DHA. But that was the conclusion from the statistics.
Mm-hmm. Thank you.
But it's true, there is so many compounds in fish, but also in algae and in marine resource, that it's complicated sometimes to just focus on one type. And I think it's something to do in the study to be able to looking more on the impact and the effect and the mechanism also of the different compounds from marine resources. I have a question for Frederick. Do you maybe talk a little more about specific mechanism of DHA on brain and on the cells?
Well, it's a bit complicated because omega-3 fatty acid tends to have different effects on different endpoints. Among those that are maybe more strong, the first one is probably the incorporation of omega-3 fatty acids in cellular membrane that can affect the fluidity of the membrane and some electrophysiological parameters. So we've seen some really basic electrophysiological parameters that were modified by the diet. So this could easily explain changes in the cognitive performance, for example. There are some evidence also that omega-3 fatty acid can alter gene expression, because a part of the omega-3 fatty acids in the membrane can go down inside the cell and modify gene expression. And there are some evidence that neurotrophic factors are affected by omega-3 fatty acids, and these neurotrophic factors have been associated with positive outcome in these diseases. There could be also there are also some evidence on oxidative stress. This is a bit more tricky because omega-3 fatty acids can be also sensitive to oxidative stress, but most evidence suggests that it's also protective. There was another one mechanism, I think I forgot. Well, yeah, but there are different studies that may suggest different possibilities, but I think those are probably the most likely.
Okay, thank you. So yeah, I think we finish here the webinar. Maybe you have some question?
Yeah, I have a few questions to Lisa, actually. The first one is really, you've shown that the kids, they had a relatively good dietary intake of omega-3 fatty acids, at least a good part of them at the beginning of the study. So I was wondering if, maybe I missed it, but in your result, did you stratify for the dietary status before the study? So maybe that those kids who had a lower amount of intake of DHA maybe did better in the test after the intake of the.
I think it was adjusted. It's a while ago, this, but it's a good comment on. But I think the project leader, she adjusted for the levels of DHA and let's see. Yeah, it was different. The compliance and of course the DHA. I think so, but it's a good comment that they might increase more in effect.
Yeah, because if you take another place in the world, I'm not so sure that you would find so many kids following the advice in terms of fish intake.
We tried to find people that were less good at following advice, but they wouldn't participate.
Yeah, exactly. So
we tried different ethnicities, but it was a very high degree of Norwegian children attending. So that's often seen in intervention studies, that those that are very focused on food, those are the ones that attend. So I think the level was quite high for all the children.
Yeah. And my other question is that in this age group, you may start to see some kids with ADHD, attention deficit disorders. So did you excluded some kids for that, or what? Maybe it was not.
No, we didn't exclude anybody. So I'm not sure how the psychologists, how they did this, but I think they tried to, for all the children, they tried to do the test. It's 1 hour, so maybe some of the children weren't able to do the whole, and they were of course not in the, so they had to do the whole test, be able to do that, and some of them got lost in the follow-up or weren't taken into the statistics if they didn't. So maybe some of those with ADHD weren't able to do the whole test.
Okay.
Thank
So,
you.
do you have some more questions for Lucy, Frederick, and Lisa? No.
Okay.
Okay. So, thank you very much for your participation. Thank you to all the speakers, Lisa, Frederick, and Lucy. Thank you very much. You can check our future events in our website from the cluster Blue Legacy. And have a good afternoon and a good day for you, Frederick.
Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Bye-bye.